I'm going to go ahead and get started with the next presentation. We're really excited to have this panel here. As Carl said, there are a group of nationally recognized experts in their various fields and have direct experience with looking at the health effects of various kinds of pipeline facility releases. So I'm going to go ahead and save your time, suggest that you read their bios in the packet because they are quite impressive. But to begin with, I'll introduce them individually as they come up. Dr. Eduardo Olaguer, who's from the Houston Advanced Research Center, recently published a paper about some work he's done in the Houston Ship Channel. And we'll introduce the other two as they come up. Thanks.
Okay. Well, this panel is about public health. And I am not a health scientist per se, but my work has focused more on the exposure side of the fence, which is incredibly important in making the link between industrial activities and actual human health impacts. So I will be talking about a field study that we conducted in February of 2015 called BTEX, the benzene and other toxics exposure. The word exposure is missing there in the title, field study. Of course, that's kind of a pun, since when you say BTEX to an environmental professional, they usually they mean benzene, toluene, ethylbenzene, and xylenes. And those compounds do figure prominently in the field study, so aromatics, but that was not the exclusive focus. So let me take you through some aspects of this study. So this was something that we did in the Houston Ship Channel and various fence line communities. Basically this was about measurement of not only ambient concentrations, but also being able to say something about sources that were responsible for high concentrations that were observed. So source attribution, as well as emissions quantification, were at the center of this study as well. Okay, so I mentioned it was conducted in February 2015. It cost us about $1 million to put together, and the funding came from U.S. Fish and Wildlife. My organization, Houston Advanced Research Center, or HARC, was actually a subcontractor to Harris County, which was the prime Harris County, Texas, which includes the city of Houston. And we had a number of other participants in the study, including UCLA, the University of North Carolina School of Public Health, and a private firm, Aerodyne Research Incorporated, which some people in the audience may know about because they're very prominent in the air quality business and the atmospheric science business in general. Okay, so I'm going to go through various technologies that we demonstrated in the field during BTEX, and we tried to make this a very cutting-edge field study. And since this is a health panel, I'm going to start with something that health practitioners will appreciate more than some other things. That's the use of cultured human lung cells as kind of exposure indicators. So this is the University of North Carolina School of Public Health. They took human cancerous lung cells and exposed them in vitro to air pollution. And what they did was that they looked at the response of cells. They looked at the enzyme and protein releases in response to ambient air. And they also did some measurements of messenger RNA. Okay, so they did some genomics too as well. And so the aim was to look at toxicity, inflammation, immune system, and cancer endpoints. And they did positive controls and also clean versus ambient air. They used ozone for positive controls. I guess they didn't have enough resources to do benzene positive controls. So what's interesting about this, and they set the lung cells up just outside a major refinery in the Houston Ship Channel. And we had also ambient concentration measurements side by side with the lung cells. And what they found was significant differential expression between clean air and ambient air. And so they found this differential expression in 10 immune-related genes and in 1 cancer-related gene. So, so that's one very significant aspect of the result of the field study, and this is already published in Environmental Health Insights, which is an open access journal. Okay. So another interesting technology that we demonstrated in the field was the use of remote sensing-based computer-aided tomography, effectively CAT scans, just like they do in medicine, okay? And the remote sensing technique that we used in the field was differential optical absorption spectroscopy, or DOAS, which is ultraviolet and visible radiation based, okay? And what's different about, now DOAS has been in use, there are commercial DOAS systems, but this is a custom built system that uses an artificial light source. And what's novel about the light source in this case is that they're light emitting diodes, okay? So they're narrowband devices that we had to kind of stitch together to get a good source spectrum. Okay, so what I have on the left here is a picture of the instrument at UCLA and Professor Jochenstutz's laboratory. Okay, and the other side of this chart shows you the light paths that were set up in Manchester neighborhood, right beside the ship channel. Manchester is a neighborhood in the city of Houston. It's a fence line community, and you can see a big refinery there in the corner. That's the Valero refinery. Okay, and so the red lines represent the light paths. Okay, at Hartman Park and at Manchester Street, we had these towers that we constructed, and the light sources and the detectors were based on the towers. And then everywhere else, you see at the endpoints of the light paths, we set up retroreflector mirrors. So the light source shines light, it bounces off the mirror, and then is aimed back at the detector at the same location as the light source. And so the absorption of ultraviolet and visible radiation in between tells you, okay, what compounds are doing the absorption and how much of the compounds there are. So on any given path, you can tell what the average concentration along the path is. And so this is on a neighborhood scale, so the typical light paths are like 1 kilometer long or thereabouts. Okay. So, but what's different with computer-aided tomography is that you can generate a lot more information if you can have several light paths that are kind of crisscrossed or overlapping, and in which case you can either use the mathematical constraints of the system, or you can use a physically based model to kind of fill in information. And you can generate essentially a concentration map that shows you where the hot spots are. So now you don't just have a single number characterizing the neighborhood, okay, in terms of what the ambient exposure is. You now have a map, and you know, if you have a heat map, you can tell, oh, this little spot here is lighting up. Something's going on in that vicinity. Okay, so now the part of the issue that we encountered in the field, it's nice in theory. In practice, there are some difficulties that you have to overcome, and one of the big difficulties that we had in implementing this technique was the fact that the trees in the neighborhood were very tall. Okay, so they blocked the sight lines, and so if you look at the light path, it's kind of an irregular network there. There's at least 2 obvious missing paths. Okay, and that's because of the trees. So it's sort of a low resolution CAT scan network, certainly lower resolution than what we originally intended. Okay, but despite the drawback, we were actually able to see some very interesting emission events, because fortunately for us, they were not in the area where the degraded resolution was. Okay, it was in the area, the northwest corner there of the Manchester neighborhood where the action was going on. I'll tell you a little bit more about that. Okay. We have a number of publications that have come out from BTEX. I already mentioned one on the lung cells. There's one that just recently came out in atmospheric environment on the CAT scanning technique, and there's another one in review that's pending, which is a companion paper. Okay, so another technology that we demonstrated in the field was the use of mobile real-time instruments. Okay. So when I say real-time, I'm talking about instruments that can measure ambient concentrations, in situ ambient concentrations, so right at a point, okay, with 1 second time response or less, okay? So you have a choice. There's no one size fits all, depends on what you want to measure, okay? So if for BTEX, we work mostly with proton transfer reaction mass spectrometry, or PTRMS. There's a firm in Austria that makes these instruments. The name of the firm is Ionicon. And so that we had 3 mobile laboratories, all equipped with PTRMS instruments, all equipped with portable meteorological stations, so you know which way the wind was blowing, and that was very important in terms of source attribution. Okay, and then we had one particular mobile lab, the Aerodyne folks. They're sort of like the kingpins in this area of real-time measurements. They had much more than a PTRMS. They had a suite of other instruments, including 1 or more quantum cascade lasers, tunable quantum cascade lasers that could zero in on things like formaldehyde and sulfur dioxide. So you could fingerprint a source more than beyond, just beyond the BTEX compounds. Okay. So at any rate, one of the mobile labs, this was one that was run by the University of Houston, was actually not mobile, it was stationary. Its job was to stay beside the lung cells and provide continuous ambient measurements near the entrance of the Valero refinery. And the others were mobile. My organization ran one of the mobile labs. It's the HARC van you see there on the right. Okay, we had less instrumentation than the Aerodyne folks, but in a way that was good, because we were like they were the battleship, we were the light cruiser, you know, so we were like the Pathfinder, you know, one of the things that was possible because we didn't have that as much data to handle was that we were able to do real-time data broadcasting during the field experiment. So what that meant was that every 6 seconds we took all our BTEX measurements and then broadcast them to the web, so that of course we didn't want any lawsuits. Okay, so we restricted access to the field investigators. And so what you see here is a web screenshot of the data broadcasting system, and the green dots are essentially the HARC Mobile Lab location, so it leaves a trail of breadcrumbs, and you see the breadcrumbs moving in real time. So you can tell where it is at any given time, since there's a GPS on the mobile lab. Okay, and on the right-hand side is both tabulation and a graph of the chemical observations as well as the wind observations. So what's selected there is C2 benzenes, which is ethylbenzene plus xylenes. Okay, and you can see there's a huge spike of, well, effectively xylenes, because ethylbenzene isn't that much there. It's mostly xylenes. So there's a huge spike of xylenes approaching 70 ppb somewhere outside the gates of the refinery. So this was on a dry run in the November preceding the actual field study itself. Okay, so it makes it very easy for someone like me who, you know, I mean, I'm to sit in the comfort of my own office and see what's going on, and I can call the operators on their cell phones and say, hey, you're seeing a really nice peak over here, why don't you move over there, you know, and upwind and or circle around this particular source. You might find it very interesting. Okay, so now it's nice to collect information in real time. You'll get scads and scads of data. Well, what do you do with it? Okay, how do you make sense of all that data? So we really thought about this well in advance of the field study. In fact, a few years before the field study even took place. And so we decided to build a data interpretation engine that would actually allow us to interpret the data in near real time, that is within half an hour to an hour of observations. And by data interpretation, I mean both source attribution / emissions quantification and plume reconstruction. Okay. I've published several papers on this. Basically, it's a microscale model that allows us to do all of this. Okay, it's an Eulerian grid model that handles both transport of air pollution and chemistry, which is quite unique. It's got its own customized gas phase chemical mechanism of 47 reactions. And you can, what's nice about it is that you can run it at very high resolution. So typically 100 meters if you don't have chemistry. If it's chemistry, maybe 200 meters. Okay. In this case, benzene, for instance, is not very reactive, so you don't have to worry about chemistry since you're very near the source. Okay, so the other thing is that the chemical transport model is coupled to a very sophisticated urban wind model called QUIC by Los Alamos National Lab. And what it does, what the QUIC model allows us to do is to extrapolate wind fields from limited measurements so that we can see what the wind field may be doing throughout the entire neighborhood, given the shape of the urban canopy. So what we did was we got LiDAR data for the urban morphology from the National Geointelligence Agency. I had to convince them that I wasn't a terrorist. And so they gave us that data. And so we constructed a digital model of Manchester neighborhood. And that allowed us to use the QUIC model to do meteorological data assimilation and essentially make sure that our wind fields that we use for source attribution didn't go right through storage tanks. They curled around and that sort of stuff. So, so you can see here the urban canopy. In Manchester, the red areas are residential and the blue areas are the more elevated structures. Okay. So what's another interesting feature of this, this model is that you can run it in the traditional forward mode, which means that you rely on emissions that are either estimated based on engineering principles or reported by the companies, which actually it's the same thing. And then you turn the model crank forward and you generate the expected plumes, the concentration plumes around the sources. Okay, that's the traditional way of doing things. But in this case, we can run it backwards using a technique called 4D variational data assimilation. It's based on the calculus of variations, and we use we build an adjoint version of the model to do this. And so you can turn the crank backwards, starting from measurements, real-time measurements in this case, and you can tell then if you see big spikes. Okay, where's that coming from? Oh, it's that flare over there. Okay. Oh, it's that group of storage tanks over there, and this is how much is being emitted, and this is when it's being emitted. So we can now study emission events, not just say how much is being emitted on an annual basis, you know, based on a wave of a hand. Okay, we can actually measure, okay, emission events. Okay. So here, for instance, is a reconstruction of a toluene plume that was associated with rail car unloading operations in the middle of the night. Okay, this was the event I mentioned that we caught with the CAT scan first. At the time, the mobile labs were not operating at night. And at around 4 in the morning, the CAT scan operators started hearing this rumbling noise. It turned out to be a train coming into Manchester neighborhood, and all of a sudden they saw this big spike in toluene. Okay, so we wisened up. We said, okay, starting next night, we're going to stagger our mobile lab operations, so there's always a mobile lab 24/7. Okay, and in this case, the HARC mobile lab was the one that caught the succeeding day's plume, identical event, practically. Okay, and they also bumped into the rail car operators. They saw them loading and unloading stuff, you know, at the appropriate emission points in the neighborhood, which we knew where they were because of the emission inventory. Okay, and so this is the reconstructed toluene plume associated with either the CAT scan, okay, on the left, or the mobile lab PTRMS measurements. Okay, so you can see that because the mobile lab can zoom in on the source, okay, it's more detailed. It's bifurcated. The core is bifurcated. The contours are tighter. The low-res CAT scan is kind of a good alert system. Okay, it kind of diffuses the plume a lot, but it gives a little. But you know, it gives you a good idea of what's going on, and then you can send the mobile lab. Okay, look at that more carefully. Okay. So, because this is a pipeline conference, I do have something related to pipelines, and it's actually the biggest story of the campaign, which got picked up by the media. And so, the time that we saw the big emission event from the rail cars, okay, that was 4 in the morning of a certain day. And then the same calendar day, after the mobile lab folks had gone to bed and then gotten back out into the field in the afternoon and evening of the same calendar day, they started to see benzene spikes right across the ship channel in a different neighborhood, in Galena Park, instead of Manchester. Okay, so I have here a map On the north side of the ship channel is Galena Park. On the south side is Manchester. And what I have there are in red, okay, the red dots are the point sources that appear in the 2011 emission inventory from EPA and the National Emissions Inventory, okay? And those are the benzene point sources, and then blue is any other point source, okay? And also, now when we first planned the study, we had no idea that Galena Park had a huge underground pipeline system. We were not refinery engineers or anything like that, but industry gave us a hint, you better look at the pipelines. So we got the database of the pipelines from the Railroad Commission of Texas and made sure to put them in our GIS maps. And lo and behold, okay, we see a big spot of benzene, the highest, almost 40 parts per billion of benzene, just as the mobile lab is crossing a refined product pipeline. So we say to ourselves, ooh, there must be leaks there. Okay, so what we did was we did our source attributions so that we were not only attributing things to specific emission point source emission points, but also to specific pipeline segments. Okay, so we could figure things down to that level. Okay, now before I get into those results, one interesting thing we found. Okay, if you look at what the spikes, when the spikes happened, and what was going on at the ports at that time. Okay, turns out that these spikes seem to be correlated with loading and unloading at the docks. Okay, at one of the port terminals. Okay. So at the Kinder Morgan port terminal in particular, if there's anyone from Kinder Morgan here, there usually is at some of these presentations. Okay, so it makes sense because you know, if you're going to see the biggest leaks, it's in the pipeline system, it's probably going to happen when you're drawing from the pipelines, right? If you're filling storage tanks after you've emptied them or whatever, you know, at least that's what some industry folks clued me into, okay? So, and we didn't have any idea of this, that there was this type of correlation that we would find, okay? So at any rate, when we totaled up the in this case, benzene, okay, from the traditional point sources versus the pipelines, okay, well, it turns out that the pipelines are emitting twice as much benzene as the point sources. Okay, this is a big surprise to us. Okay, and now another interesting thing about these pipeline leaks that we saw is that when you actually quantify this down to the segment level, the pipeline segment level, what we see is the most we see is like 3 kilograms per hour. You know, that's less than a gram per second. So that's less than the detection limit of an infrared camera. Okay, so what that means is that your even though industry flies infrared helicopter-mounted infrared cameras to detect leaks, they're not going to see any of this stuff, and yet you will see 40 parts per billion concentration peaks as a result of these types of leaks associated with the loading and unloading. Okay, all right. So here's a reconstruction of the benzene plume. One is the early afternoon, and then the second is the evening, the two different peaks that we saw. All right. So health-wise, what does this mean? Okay, I'm not a health scientist, but TCQ gives some guidance, although they're not as conservative as other agencies. So they have what's called an effect screening level for various toxic compounds. Okay, and they have a short-term ESL for benzene of 54 parts per billion, and a long-term, usually annual mean, but of course in practice you don't make annual mean measurements, you make 24-hour measurements, typically with canisters and whatnot. So that long-term screening level is 1.4 parts per billion. So we're definitely approaching the 1 hour ESL for benzene.